All right, so welcome everyone to our SNEV special series talk. I'm Swagatama, and we also have Natalie joining us for the session. So today we have Dr. Keerthie Dissanayake with us, who will be talking about his journey with EV research and existing challenges and bottlenecks in the field, especially in Sri Lanka. And Dr. Dissanayake, before he starts with his talk, I wanted to introduce him a little bit. He is a senior lecturer in the University of Peradeniya in Sri Lanka, which is also his alma mater and also the place where he got his degree in medicine from. He then moved on to pursue his doctoral studies with Professor Alireza Fazeli's group from the University of Tartu, Estonia, where he investigated the pre-implantation embryo-derived EVs as an embryo quality marker and the role in a mediator of embryo-maternal communication. So without further ado, I request you to begin the talk.
All right, thank you very much, Sugatama, for the kind words of introduction. First of all, let me thank the organizing committee for inviting me for delivering this speech at the student network for extracellular vesicles. So especially the Dr. Noor Hayati, one of my friends from Malaysia, she's the one who suggested me. So thank you very much, Noor, if you are around. Then all the other organizers of the group for inviting me to deliver this speech. When I was given this task, the biggest problem that I was having is how to link two entities. That is the scientific part that I had to talk about, and this the sociological issues and others that are there when it comes to starting EV research in Sri Lanka. So that's why this the link of the combination of these two areas within my study, the presentation title. So hello everyone. Simply this will be my story. This will be my story for the last 5 years, that no more no less. So it would be please bear with me for the next 45 minutes. I'll try to be confined to the time limit during my presentation. Well, it was year 2017, the mid-year 2017, that I was while I was working as a probation lecturer in anatomy at the Faculty of Medicine, which was the same school from which I graduated. I was completed my master's. Then I was looking for my PhD position because that is the high time for that. Since I was teaching undergraduates on embryology, I was so passionate about embryology, so I was looking for a possible location to study or pursue more further studies in embryology. Then I was introduced to a particular gentleman in UK whose name was Professor Alireza Fazeli, by one of my colleagues and mentors from the University of Peradeniya, Professor Suranga Kodituako. At that time, Professor Alireza Fazeli was not only the professor in reproductive medicine at the University of Sheffield in UK, he had been recruited by the University of Tartu under a transgenome project to serve as the ERA chair of translational genomics at the University of Tartu to run this particular project, which was funded by the European Union's Horizon 2020 project. So I was discussing with him, and he introduced me. Then he interviewed me and recruited me, and he offered me the opportunity to study for my PhD at the University of Tartu. But originally I wanted to go to UK for my certain personal reasons, but based on these discussions, then I realized that I can do part of my PhD in UK. So I was very happy to go to Estonia to start my PhD. So this is the Estonia within the European mainland. It's a part of Baltic region, which is neighboring with Finland, Sweden, and on the right side you will see the Russia. So this is a small country with a group of very nice people. So I ended up in this beautiful country. I didn't go alone there. I had one of my partner in crime. So he is Dr. Kasun Godakumara. So we had the same schooling, and then at the same school, then we had the same university undergraduate and postgraduate education up to the masters. So we went on this journey to reach Estonia for the first time. That was my first visit to the Europe. So we went to the Estonia through Turkey. So we were kind of potential illegal immigrants at that time when we were traveling to Estonia, because we had no return ticket, so we were heavily questioned, but still we ended up in Estonia. So finally we came to this wonderful place called the Faculty of Medicine, the Institute of Biomedicine and Translational Medicine, University of Tartu. Professor Fazeli welcomed us, And he explained to us clearly the scope of this project and the limitations and the restrictions and all very clearly at the very beginning. So people are still getting on board for the project. So it was still at the very beginning. So I was introduced and I was told that we had to work on EVs. Of course, beforehand I had knew that. So working with EVs, of course, to be honest, I had never heard of that EVs as an undergraduate level or a master's student. So what is the fascinating thing about EVs? I think many of you who in this audience be knowing that. The task given upon me was isolating EVs from embryo conditioned media. I was very happy when the task was given simply because I wanted to learn in the field of embryology. So of course I was very happy at that point, but for a moment I was unhappy as well. The simple reason was it is extremely challenging to isolate EVs from embryo conditioned media which is in microliter quantities. So it would be different from isolating EVs from cell culture media, blood and other biological fluids which are having loads of EVs. So that was the task at the beginning. So what is the purpose of isolating EVs from embryo conditioned media? That is the biggest question. Then with the discussions, we realized at the end there's a clinical condition which is called infertility. That would be the target situation that we would be heading towards. Looking for the answers for. So infertility, as you all know, is a global health problem, right? So the WHO, the World Health Organization, have defined infertility as the third most prevalent disease in the 21st century after the cardiovascular diseases and cancer. It has made a huge impact to the affected families and certain communities, as you all know. There are different types of management modalities, including the simple medical treatments to surgical interventions and also the assisted reproductive technologies. Now, when you say assisted reproductive technology, you know it's about IVF simply. So during during assisted reproductive technology, the gametes, or especially the oocytes and the embryos, are handled outside the mother's womb, and then they are manipulated in such a way that finally a good embryo is transferred to the expectant mother for the continuation of the pregnancy. The two key findings, if you look at the assisted reproductive technology and its history, it's a fascinating story. I found out two key stories. One is about the 1826, when the Carl Ernst von Baer, the professor, German Baltic or rather Estonian professor, who identified this ovum outside the mammalian embryo for the first time. He identified this mammalian embryo that can be considered as one of the turning point of this story. Subsequently, because he is considered as the father of embryology as well due to his various identifications and definitions, including the explanation of germ layer theory of embryology. There were many success stories, there were many stories, and finally, in 1978, the birth of the Louis Brown, the first IVF baby, took place in UK, and that was a revolutionary incident, and all the world opened their eyes towards this incident. And then the IVF technology and everything revolutionized, and it revolutionized the management of the infertility. Well, now currently, despite these advances, still there are very many challenges for the for the best results. So two challenges are the identification of the embryo quality and identify the embryos with best quality and predicting the transfer success. So the other one is the assessment of the endometrial receptivity. The reason is you may transfer the best quality embryo, but still they may fail to implant in the mother's womb. So these are the two key areas where more further research is needed. When it comes to the embryo quality evaluation, you know, the morphology-based embryo quality evaluation is the key. This can be considered as the gold stone, but still the gold standard method, but still it has its own problems, right? In addition to that, the time-lapse micro imaging based this morphokinetic evaluation of the embryo quality is another possibility. Then when it comes to the non-morphological method, the pre-implantation genetic testing, which is an invasive method, it cannot be utilized all across all the embryos to evaluate. So there are certain very many limitations. Let's come to the analysis of the embryo conditioned media. Currently, there are so many publications who have tested the cell-free DNA, non-coding RNA, proteins, metabolites, oxygen consumption, all these sorts of biomolecules to see if they can be used as embryo quality markers. On top of that, our research group and Alidisa had this potential question: if extracellular vesicles, can they be used? What are the potential implications in embryo diagnostics? That is how the area where I research come to the real clinical story. Well, I don't have to tell you what are EVs. As you can see, EVs are membrane-bound biological nanoparticles which are secreted by almost all types of cells under different pathophysiological conditions. So they carry a molecular cargo, and these EVs can reflect the physiological status of the releasing cells or the pathological status. Based on that, these EVs can be considered as potential biomarkers. Similarly, in the extracellular hostile environment, these EVs are very stable. Because of that, they can be considered or developed as potential therapeutic drug delivery systems. So these are two key major areas where the future of EVs are there in the field of biomedicine. In addition to that, you all know that these EVs, once released by these cells, can be internalized by their target cells. And these target cells, in the target cells, they can alter the phenotype, right? It can be in different forms, but the phenotypic changes can be done. So these are the key features of EVs in brief. In 2018, when I started this project, that isolation of EVs from the embryo conditioned media, there were only a couple of papers that had been published who had worked on grouped embryo cultures, both in human and bovine, and they had proved that embryos, of course, release EVs. And then, of course, they showed that the embryos can internalize these EVs. So it's a bidirectional flow, right? So that had been shown, but it's a very limited, only 2 or 3 papers maximum. Based on that literature review and our idea that if the cells can reflect the pathophysiology of the releasing cell, then of course we thought that then the embryos also can reflect the embryo quality. That is a kind of an hypothesis that we had at the beginning. So in my first study, I wanted to isolate EVs from the embryo conditioned media and then subsequently characterize them and to see what kind of differences are there in EVs based on the type of embryos that they were released from. So in this study, what I used is bovine embryos as a model for this human condition that I'm talking about. So initially, I'll go through briefly the study design. So initially, we cultured embryos in groups, as you can see here. And then after the IVF, this is potential, the main mode of making embryos was the IVF, the conventional IVF. And then the presumptive zygotes subsequently were cultured individually as droplets. This is called droplet culture. Then they are separated from each other under the mineral oil. And they were cultured for 8 days, up to the day 8. And during this time frame, they were evaluated morphologically at day 2, day 5, and day 8. But from different batches, we collected a fraction of the conditioned media at day 2, day 5, and day 8. And collected fraction was stored. However, the embryo were allowed to grow in the remaining media up to the day 5 and day 8. So at each point, we recorded the development of these embryos, and then we collected separately the conditioned media. Retrospectively, we label the collected conditioned media as day 2 good quality media, day 2 bad quality media, similar for the day 5 and day 8, depending on how the embryos reach a 2-cell stage at day 2, morula stage at day 5, and the blastocyst stage at day 8. Right? So subsequently, the biggest challenge for us was isolating EVs from 50 microliters of embryo conditioned media, and we knew that it's going to be a challenge. At that time, our group had started isolating EVs using a high-performance liquid chromatography machine, which is originally a method used for the protein purification, and this is the size exclusion chromatography column used for that purpose. And I tried to isolate EVs from this method. Because the good thing about this method is it has a chromatogram which is being displayed on a computer screen, so we can see when which fractions the EVs are coming or in which fractions the unwanted proteins. Now when it comes to size exclusion chromatography method, you generally can get two peaks. One is the first peak which technically can get the EVs, and the second peak which will be the unwanted proteins and all. So we tried with this method for some time, for maybe closer to a year. But there were so much of intergroup and batch variations, and we failed to characterize subsequently with electron microscopy and Western blot based proteomics and all. Simply we know that we had a limited amount of proteins from embryonic EVs, not like cell culture media and all. Therefore, we had to change our plan. So we shifted to another method, which is a benchtop size exclusion chromatography column, which is a product of this particular company. And it changed my whole story. And in this method, we were able to isolate and subsequently validate the EVs. Now, this is one of the instruments that is there in the facility lab in Tartu. This is called the nanoparticle tracking analyzer, and we consider this as the heart of the lab, because all our EV experiments are finally end up in this position before moving towards the other experiments. So we quantify here both in the scatter mode and the fluorescent mode to characterize our EVs. So we characterize the EVs from this method, and then subsequently with electron microscope, not only transmission electron microscopy and also the scanning electron microscopy. Still, the issue was that both the embryonic EVs and the control media both had nanoparticles. Now EV depletion from the culture media is a key step in the EV research. But when it comes to the embryo culture, it is not possible. If you deplete that particle, then the embryos won't go beyond the morula stage. That is one of the findings that we had. Therefore, we had to keep them in the background. However, when we perform this EV array, the EV array is a technology which is an antibody-based method developed in Denmark by Professor Malene Jorgensen and her group. So thanks to that group and with the collaboration with them, that we were able to prove that embryo conditioned media derived EVs contain nanoparticles, whereas nanoparticles isolated from the control media were not containing or not positive for these markers. Therefore, we concluded that embryo conditioned media carries EVs which are actually released from the embryos. So that was that took place after 1.5 years after starting the project. I think that is a kind of a big event in the course of my study, because this getting positive results for this one, this biochemical characterization was the biggest win in the whole story. Subsequently, once we proved that we are having EVs in our embryo conditioned media, we tried to compare based on different characteristics. Now, physical characteristics of EV is one way, because in other terms you can try to do the proteomic characterization comparisons, then the RNA based comparison and all, but depending on the limited amount of materials and resources we had in that case in terms of EV amount, we just compared these things based on the particle concentration. Right, that at day 2 conditioned media samples, the nanoparticles between the bad quality embryo media and the good quality embryo media were different. Now, mind you, at this time in these both groups, the morphologically the embryos were similar, but in the bad quality embryo media, after cleaving at day 2, the embryos subsequently degenerated, whereas in the good quality group, the embryos were developed to the blastocyst by day 8. Therefore, whatever which was not visible morphologically at day 2 has been shown based on this quantification. Similarly, at day 8, we observed that between the bad quality and good quality embryo media differences are different. Now, this was evaluated based on a linear mixed model. So, these differences were the initial finding that we observed in terms of concentrations, but similar differences were not observed at day 5. Moving to the comparisons of the average size of these nanoparticles or EVs, we observed that at day 2 the particles were larger in the bad quality embryo media and the good quality embryo media samples compared to the control. However, between these two groups there were no differences. If we take day 8, of course the size was smaller if you compare it to the two sides, there was a difference. Another thing I wanted to mention here in terms of concentration is bad quality embryo media had a higher particle concentration compared to the good quality embryo media at day 8. Now you know when the cells are stressed, when cells are dying, maybe they are releasing more and more nanoparticles or EVs to the culture media. That may be reflected in this study. So finally, after 2 years and 3 months, or 2 years and 2 months after starting this particular project, we were able to publish our first study in Theriogenology, titled as Individual Cultured Bovine Embryos Produce EVs that have the potential to be used as non-invasive embryo quality markers. Now this study is a good example of collaborations, because thanks to several collaborations that we had, especially the Alisa facility had, we were able to network with different groups across Europe to make this study, to bring that study to its end. So otherwise, at the very beginning, we had limited technology among us, so otherwise we would have faced trouble. Now moving to the second study, now as you all know, the fertilized the oocyte, then the zygote, moves along the maternal tract. They become 4-cell stage, the 8-cell stage, then become the morula, and then reach the uterine cavity for the purpose of expecting to be implanted in the maternal womb. Now, studies have shown that during this journey, the embryo communicates with the maternal tract. Now, this we consider as the first. So we consider as the first level of communication between the mother and the baby. So we suggest that EVs are the first language of this communication. Now here in the second study, what we wanted was to see that if the EVs coming from a particular cell can affect the phenotype of a recipient cells, then we expected that EVs coming from the embryos can affect the gene expression of the maternal tract. Here what we did was, similar to the first study, we cultured embryos individually, and depending on the embryo quality, we collected separately the embryo condition media as good quality embryo media and the degenerating or bad quality embryo media. Then separately we isolated EVs, and these EVs were supplemented to the primary bovine oviductal epithelial cell cultures, expecting EVs from these good quality embryo media and the degenerating embryo media affect the gene expression of these primary bovine oviductal epithelial cells. Similarly, we supplemented as a control nanoparticles isolated from a control media sample, or the conditioned media, which were conditioned without any embryos. So they were also in the incubator for the same time point. So we had 3 groups: good quality embryo media EVs, bad quality embryo media EVs, and the control. Now these controls, then subsequently we collected the cells, the monolayers from the primary bovine oviductal epithelial cells, and then we use them for RNA sequencing, differential gene expression analysis, and the qPCR for subsequent validation. We had 4 groups, 4 biological replicates. Probably because of that reason, we have a huge amount of, I would say, intra-group and intergroup variation across these samples. However, we did this subsequent differential gene expression analysis, and as you can see, the good quality embryo EV group versus the control, the degenerating embryo EV group versus the control, and then the comparison of the 1 and 2 groups. Interestingly, for the first comparison, we didn't get much number of differential expressed genes, but 7 upregulated and 18 downregulated. Similarly, for the third comparison, similar number. However, when the bad quality embryo derived EVs were supplemented, we didn't get any number of significantly enriched differential expressed genes. As you can see here in this graph, you can see the first comparison and third comparison, many of the genes were overlapping, as you can see here. Among these differentially expressed genes, among these upregulated genes, you can see OS1Y, MX1, and ISG15, and this gene locus. All these are, when I went through the literature, I realized that these are genes now referred to as interferon stimulated genes, or genes belonging to the interferon tau pathway. Now, interferon tau pathway is something very important in the ruminant pregnancy, because similar to the humans HCG or human chorionic gonadotrophin in human pregnancy. Interferon tau is the pregnancy recognition molecule in the ruminant pregnancy. So possibly the good quality embryo derived EVs must have carried this interferon tau, which is the reason for the upregulation of this these upregulated genes. We further validated these findings using the qPCR, but we got only good results for the OS1Y and the MX1, but this locus was not positive in that particular result. It didn't go in line with the differential expression RNA sequencing data. So with these findings, what we proposed was good quality embryos, when they come into contact with the maternal tract, here in this case the oviduct, they release EVs, and these EVs are internalized by the maternal tract, and then they alter the gene expression of the maternal tract. That means possibly the maternal tract can sense the embryo, right? Not only sensing the embryo, its quality as well. So this work was published in 2021 in the Journal of Molecular Medicine, where we proposed that we concluded that the pre-implantation embryo-derived EVs alter the gene expression of oviductal epithelial cells, and such altered gene expression is dependent on the embryo quality. Now, moving from the oviduct, the embryos reach the uterus. Now, uterus is the purpose of embryo reaching the uterus is the implantation. Now, this is the most important step in the whole process of pre-implantation embryonic development. It has three phases: the apposition, attachment, and finally the invasion. Now, for this to be successful, there should be a good quality embryo, there should be a receptive endometrium, and there should be a dynamic optimum communication between these two entities. Now, understanding the EV-mediated communication at this embryo-maternal interface is not possible in vivo. That is why people are using different types of cell culture-based models. Now, in this purpose, here at the facility lab in Tartu and even in the Sheffield, the researchers are using a model where the trophoblast or the embryo is represented by spheroids which are formed of the human choriocarcinoma cell line, which is called JAR. Similarly, representing the receptive endometrium, another cell line is being used, which is called RL95 cell line. So, using these two cell lines, a study conducted in the same in Tartu, led by Masumi and Kasungoda Kumara. They were the first authors in this paper. They showed that the EVs released by these trophoblastic cells, or trophoblasts, they can transfer these transcripts, SNEV81, this exonic region and this intronic region, all through the EVs towards the maternal tract, that is to the RL95 cells. And subsequently, this supplementation leads to the downregulation of the same transcripts in the RL95 cells. And this is one of the consistent findings that we observed with qPCR based method was that if you supplement JAR EVs to the RL95 cells, the SNEV81 downregulate. Now, because based on this model, we further continue to use this downregulation as a marker when we are studying the EV-mediated embryo-maternal communication. Now, after identifying this communication, we came to this question that to which extent the EV-mediated embryo-maternal communication is specific. Now, this is a very broad, widely studied area in EV research, as you all know. The specificity of EV, intercellular communication mediated by EVs. How specific? There are studies to support that it's specific. There are studies to support that it is not specific. However, in terms of embryo-maternal communication level, we wanted to explore this further. Now, here what we did was, we have these jar cell spheroids representing the trophoblasts of the embryos, and then the RL95 cells. We simply wanted to supplement EVs from these embryos to these RL95 cells and to see how this affects the gene expression of these epithelial cells using RNA sequencing. So this is the study design here. Primary jar cell cultures, jar cell monolayer culture, the spheroid formation to represent the trophoblast spheroids, and then subsequently the released EVs were collected and then isolated EVs. As a control, HEC293, a human embryonic kidney cell monolayer was cultured, spheroids were formed, and the released EVs were isolated. Conditioned media were collected and EVs were isolated. And these EVs were separately supplemented to RL95 cells. JA EVs here, HEC293 EVs here, and then the third one was RL95 cells alone without any supplementation, which was the control. So all these monolayer cultures, monolayer cells, cell monolayers, are subsequently used for RNA isolation, mRNA sequencing, and then the differential gene expression analysis. So we wanted to test if is it only the JA cell derived EVs that can affect the gene expression of RL95 cells? What about another EVs from another source? So the primary question is how specific this communication. So we had these three groups. Then of course what we observed was that the JA EV supplemented group, despite it has some amount of intra-group variation, it was deviated from the unsupplemented group and the HEC EV supplemented groups. They were clustered together. Therefore, we only observed differential expressed genes in the JA EV or the trophoblastic EV supplemented group compared to the control, indicating that it's based on the used cell lines that this communication is kind of specific. Further analysis using the pathway enrichment analysis with the GSCA, we observed that some of the pathways, there were several enriched pathways, among them extracellular matrix organization and signaling by G-curve protein coupled receptor pathways. They were the kind of enriched pathways. And these are important when preparing, optimizing the maternal tract or the endometrium for the impending implantation. Well, this work pioneered by Kasun. It was published in the same year 2021, titled Trophoblast-derived extracellular vesicles specifically alter the transcriptome of endometrial cells and may constitute a critical component of embryo-maternal communication. Research continued towards the third year, and then the next question that was raised is, now we were still investigating the specificity of EV-mediated embryo-maternal communication. Now the simple question was, can only the mothers respond when embryos talk? Therefore, we want to check when the embryonic signals are passed, is it only the maternal tract that reacts? That is the simple question. Similar model, but the difference was that we cultured, we produced JA spheroids and the HEC293 spheroids representing trophoblast EVs, representing to collect trophoblastic EVs and non-trophoblastic EVs, and these EVs were supplemented there as a control as well, supplemented to 6 different cell lines. 3 were representing endometrial epithelial cells, which include the RL95 2 cells. These are our signature cell line in terms of endometrial epithelium. Then the Ishikawa cell line, another receptive endometrial epithelium. Then the HEC1A cell line, HEC1A cell line, which is considered as a non-receptive endometrial epithelial cells. Similarly, we have another control group where non-endometrial cells were used as the recipients of these 2 sources of EVs. FHC cells, which were the fetal colonic epithelial cells, HEC293 human embryonic kidney cells, and the HCC44 cell lines. This is a kind of an pulmonary lung adenocarcinoma, the small cell lung carcinoma cell line. Whatever it is, they were kind of control. Now what we wanted to do is supplemented these EVs to these cell lines, right? And then incubated for 4 hours and 24 hours for different sets of culture dishes. And then after these time points, we wanted to assess to which extent this communication was specific. Here what we used as a marker was the downregulation of selenium-81 in all these cell lines in response to the supplementation of trophoblastic and non-trophoblastic EVs. So we used absolute quantification in this respect, and then subsequently did the data analysis. So these are cell lines that we used for your information. Interestingly, if you take the endometrial epithelial cell lines, this selenium regulation was observed only in the RL95 cells at 4 hours and 24 hours. In Ishikawa cells, at 4 hours. That's all. Even at the Ishikawa cell line, which is considered as a receptive endometrial cell line, 24 hours we didn't get a statistically significant downregulation. Now this cell line, non-receptive, HEC1A, here also we didn't see the downregulation of SNEV81 copy number when the JAR EVs or trophoblastic EVs are supplemented compared to the control. There nothing was, even though we see a trend, it was not significant. So I was talking about the JAR EV supplemented, but even the HEC EV or the control source of EV, their supplementation also didn't downregulate the SNEV81 in the recipient cell lines. Non-neutron cell line, none of the cell lines were positive for this downregulation story. Therefore, in this story, we concluded that receptive endometrial cell lines, but not the non-receptive endometrial cell lines or the non-endometrial cell lines, showed the downregulation of SNEV81 expression in response to the trophoblast JAR or the JAR cell-derived EVs. So this work is further supplemented with some proteomic studies, and we are in the process of finalizing the writing of the manuscript. The last story is about a kind of a bench to bedside story, and this was a major work done by Kasankoda Kumara, and I also had the opportunity to contribute to this. And here what we wanted to do is to quantify the embryonic signaling. Now I told you at the beginning that initially what we wanted to do was to quantify the embryonic EVs to assess the embryo quality. Then at my second study I showed you that how the oviductal EVs are reactive to the good quality embryonic EVs compared to the bad quality embryonic EVs. Similarly, Kasun's primary work with this mainly the cell lines and all initially showed that SNEV81 is a good candidate marker to evaluate how the downregulation of SNEV81 takes place in the recipient cell line, the RL95 cell lines, in response to the embryonic signaling. Now this was further validated or further developed through this particular assay where instead of JAR EVs we got down conditioned media samples from the real human embryonic conditioned media from IVF clinics. Especially for this particular study was done from using a fertility center in the fertility hospital in one of the two east, I can't remember exactly, the east or the west Tallinn Central Hospital. We got down embryo conditioned media, and then they were transported along with all the other characteristics of this embryo conditioned media. So after the end of embryo culture, either they were transferred or else they were vitrified. So whatever this conditioned media is generally discarded. So we got down these conditioned media, and then we supplemented this conditioned media which carries these EVs to the RL95 epithelial cell monolayer cultures, and then the differential expression of this SNEV81 was quantified using RT-qPCR based method. Now these findings were compared with the general embryo quality evaluation method, which is the Gardner's blastocyst grading system, because in the hospital clinics still people use this as a kind of a gold standard, where they look into the blastocyst expansion, the inner cell mass quality, and the trophectoderm quality to evaluate the embryo quality. They put them together and provide a grade. So we use this, we had these characteristics with us. So then this was compared with our SNEV81 downregulation. Interestingly, this was analyzed: blastocyst size, trophectoderm quality, the ICM quality. None of these characteristics, then put them together, the overall embryo quality, none of these characteristics, morphological characteristics, were correlating with the SNEV81 expression. That means the downregulation of SNEV81 was not correlating with the morphologic characteristics of embryos. That means that whatever we observed with SNEV81 is something which is different from the embryo's morphology. Therefore, there are some elements, something called functionality, which is reflected through the EVs, which is not depicted through the simple appearance of these embryos. Subsequently, when the those embryos were transferred, then the transfer outcomes were obtained. Then, using a particular model, Cousin evaluated all these SNEV81 copy numbers with the embryo transfer outcome. Then, of course, it was observed that based on this particular cutoff level that was derived based on this particular model, that if you use this particular copy number as the cutoff level, that if copy number drops below this particular point, we can consider it as it is a good embryo for that to be transferred, and if it is the copy number is higher than this one, then that particular respective embryo is not suitable for transfer. If you use that determination of if that decision making for the analysis based on the real outcomes, then we could have told that this model would decide with a sensitivity of 78% and specificity of 83% that which is the best embryo to be transferred. So this is a kind of retrospective analysis based on the already embryo transfer outcomes. So this is a kind of a dimension which is further developing still. That where the early the bench level research can be moved towards the bedside, I would say bedside translational research. So this work should have been continued, but unfortunately I am not there anymore, but I'm sure Alisa is, and the cousin especially, will be continuing this work further. So put them together with the last study, I would say that the embryo-induced transcriptomic alterations didn't correlate with the embryo morphology. The degree of downregulation that SNEV81 downregulation correlated significantly with the outcome of embryo transfer. So I would say that the evaluating the functional competency of the pre-implantation embryo could be a reliable method of when predicting the embryo transfer success. Putting all the scientific work that we did in Tartu together, I would say that physical characteristics of physical pre-implantation embryo-derived EVs are distinct based on the embryo quality, and differential and specific responses of the maternal tract epithelial cells to the embryonic EVs indicates that these EVs carry a specific and unique molecular cargo. This was subsequently proven with some of the models actually, using some RNA sequencing and the proteomic analysis. Then again, the quantification of the embryonic EVs or response of the maternal tract epithelial cells to such embryonic EVs can be developed as a non-invasive and objective method of assessing the embryonic quality. Therefore, these kind of methods would potentially enhance the accuracy of embryo selection and transfer success in the ART in the future. So that simply concludes my scientific part. The original studies that I contributed to in the theme of embryology and embryo-maternal communication. There are several other studies that I contributed during my stay, which are not in this exactly in this field. When COVID hit us badly, right, lab was closed. So not only the original research we continued, we were able to, as a group, we were able to publish in, we were able to write certain protocols which are published in this Methods in Molecular Biology series. As I hope you have heard about this series. So we were able to publish certain protocols in terms of EV isolation and characterization. Furthermore, I was able to contribute to several review articles which were published mediated by the Facility Lab. So there were also some of the outputs that were there when I was in Tartu. In addition to that, another good work that was done there was application for this international patent titled In Vitro Fertilization Embryo Viability and Quality Assay. Now this was still under the processing. So this is another end where the Facility Lab worked for applying and getting down patents. The work in Tartu was not confined to wet lab research. We also wanted to disseminate what we learned. One of the methods that we did was this particular research symposia, research schools and EV symposia. if I am not wrong, starting from 2019, we managed to conduct research schools as well as research symposia, facilitating those who are interested in engaging in EV research. We provided a good platform to come to us and learn how to isolate EVs and how to characterize EVs. So that was a very successful story of the facility beyond research. So you can see some of the participants listening to practicals, and then this is at the end of one dinner after the end of this symposia. And here you can see Professor Malin Jorgensen. She is presenting about her EV array technology to the participant. When the COVID hit us badly, we went online. So it was more successful than the real on-site work, because we had more participants now across all the continents, if I'm not wrong. So we video recorded all the practicals and we shared with these participants, and that was one of the good work that we continued. And even if after I left, still now the facility lab is continuing to deliver and cater the scientific community to helping them in learning about EV research. However, in 2022, March 1st, exactly 1 year back, I defended my thesis, right? I defended my thesis titled "The Pre-Implantation Embryo-Derived Extracellular Vesicles, Potential as an Embryo Quality Marker and Their Role During the Embryo-Metal Communication." It was kind of that I had to finish, because I could have continued, to be honest, but there are people who are waiting for me at home, so I had to come back home early. So I've completed with some unfinished work. I returned home in 2022 March. I had so many optimistic thoughts that I would continue more and more research with facility lab. Of course, I had some collaboration, so I contributed in some other way, but that was not up to the expectation. So 1 year back I came and I said I had to say goodbye to this wonderful family. By that time the transgender family had further expanded when Alisa Fasli got another grant at the Estonian University of Life Science, which was called Combivit. So that helped us as well. So it further expanded now. So we had it became a big team. So many of us enjoyed this company. So it was a big team. So I had to say goodbye to this family and I had to leave, whether I like it or not. Then I came to Sri Lanka. Then it was the situation in Sri Lanka. It was not the country that I left in 2018. COVID hit country, worked towards economic collapse and bankruptcy. When I came to Sri Lanka, long queues for the fuel, people were striking, there were long queues for the gas, right? The foreign reserves were empty, no money for purchasing essentials. The no tourism, because Sri Lanka heavily rely on tourism. No tourists, the airports were kind of empty. Now what about EV research? This is a fight for survival. So, but still, I resumed the duties as in the same university where I was working beforehand, because I was on study leave. So I was promoted to the senior lecturer, and then I wanted to continue EV research. So I was looking for EV research. At least, is there anybody who is working on EV research? I was looking, I was contacting several people. I couldn't find any. When I do Google, the only thing when I typed EV research in Sri Lanka was this. EV, that is called electric vehicle. This is the only product that is, this is a product of 100% Sri Lankan product. It's an electric vehicle that was launched a couple of years back. So this is the only EV that is there in Sri Lanka. So I had to start from the beginning. I had to introduce EV research, because otherwise nobody is working in EVs. What was the kind of strength if I were to start EV research? The biggest strength that we do have is we have enough patients and clinical materials. That is the only advantage I do have here in Sri Lanka compared to Estonia. Estonia is a small country with 1.3 million population. If you want to get an adequate sample number, then it would be a heck of a lot of trouble, because small amount of patients that are there, simply because it's a small country. But here in Peradeniya, I have 3 teaching hospitals with huge bed capacity within just walking distance. So that is the only advantage I do have if I want to start EV research in Sri Lanka, because I have not finished my work which I left at home in Tartu. So this is, once I finish that, then I have to think of starting EV research in Sri Lanka. Now, what are the challenges of starting EV research? Now, the biggest problem for me at this point is ever increasing teaching workload. When the people are leaving, when the brain drain is happening, workload is increasing for us as academics. So, as academics, there should be a balance between teaching, research, and administration. But my work is more or less cornered towards teaching. That's why I'm kind of in a situation where I cannot spend much time for EV research, but I have to do that, because to survive in academia, I have to do research. Then, of course, compared to the 10 years back, in Sri Lanka, the research culture has improved, but still it's not good enough, because those who excel in research generally are being absorbed by foreign universities. That is a problem, because currently there are about 30, I can't remember exactly, but more than 15, the world's top scientists still remaining in Sri Lanka. But still, you never know at which time they are leaving. So therefore, networking among locals is a bit of a problem in this regard. Then, of course, to stop the corruption, there are so much of financial regulations that are being laid, and these financial regulations are discouraging if somebody wants to continue in research. That is another bottleneck. If you want to do some epidemiological research which doesn't consume much of these finances, of course, apparently that's fine. But if you want to do wet lab research which involves a lot of procurements, lot of purchasing, then of course it's a kind of a problem. Then the brain drain, that is the biggest problem at this moment. Whoever you want to trying to leave the country, that is the biggest issue. I don't know in another couple of years what will be the situation in Sri Lanka in terms of intellectuals who would be returning. I don't know. That is the biggest problem. That is why I'm holding this, saying that stop brain drain, right? Otherwise I may end up in either Estonia or wherever, the UK, Australia, wherever. So that is the biggest problem. Then finally, the inflation. If you want to buy something today, then another couple of days time, the price would be very different. So that is, inflation has hit all across the globe, but this is a big issue in Sri Lanka. These are the major issues that I am facing as challenges if I want to start EV research. Of course, they are not confined to the EV research, but to research in general. What are the alternative methods that are there? One is that, of course, if I can collaborate with certain industry-based research, then of course there are certain opportunities, because then the industry is a business. That means there I can come up with certain companies that they would be willing to provide finances for pursuing research. That is one entity, because all the other grants are drained. Then foreign collaboration.
Keerthie, sorry, we might just need to finish up shortly, so we have time for questions, okay?
This is the last slide, yeah. Then foreign collaborations and networking for EV research. And finally, the next level of research that I have not started is pursuing systematic reviews or meta-analysis. I know that there are ample amount of EV related publications, original research there online. So if you can go through and identify good research questions, this is the next level of research where I can sit down in front of my computer and conduct. And let me thank that concludes my main story. And let me thank these funding resources, Transgenome Project and the Combivate from the European Commission and the European Union, and then the Dora Plus that facilitated all my work during my stay when I was in Tartu. And finally, let me thank Professor Alireza Fazili and all the other supervisors and my fellows in Tartu who were very inspirational for me during the conduct of my work. Thank you very much. And I took more than 10 minutes beyond the time limit of 45 minutes, and I'm sorry about that. And that concludes my presentation, and I'm happy to take questions.
Thank you so much for such a wonderful presentation, Keerthie. That was really good scientifically, and then your story about going back to Sri Lanka and trying to really start things there, and obviously not the most optimal conditions. You just a really great example of what you can do when you are determined. And yeah, my hat is off to you, trying to isolate EVs from embryo culture media. I can't even imagine what you went through to get to where you are now. So well done. So I actually had a question myself earlier just to start things off. I think I missed out a couple of words in the question. And it's way back when you're looking at the particle concentrations of the day 2 versus the day 8 embryos. And I thought if you compared across the good versus the good across the day 2 to the day 8, and the bad versus bad, it looked like the particle concentrations didn't actually vary too much. And I thought that was interesting, thinking as the embryo develops, more cells are dividing, probably more things being released. It was interesting that that number didn't change too much. And then it got me thinking about the day 2 good embryos having fewer particles in the media compared to the bad day 2. And I wondered, is it something that's happening with the embryo that they're actually taking up more EVs than the bad ones? And this is why you get that deficit compared to the control and the bad. And I just wanted your thoughts on that.
Yeah, thank you very much, Natalie. Exactly that. These are the same these are the same observations we made. Let me take the first question. The changing of the nanoparticle or the EV concentration across time points, day 2, day 5, day 8. Apparently, we didn't observe a statistically significant difference because if the EVs are continuously releasing EVs, then we expect that it to be increased. But however, what is actually happening in the embryo culture media microenvironment is, while EVs are released by the embryos to the culture media, some of these particles which are existing in the embryo condition media are absorbed, internalized. So there's a kind of a dynamic exchange of particles. As I told you before, in this microenvironment, the culture media carries certain amount of nanoparticles which are not EVs, that forms the background. So that's why comparison across time points is not a good idea, because these nanoparticles are kind of forms a platform, and then while comparing the day 2 good quality and day 2 bad quality, then of course there's a difference. That means irrespective of the release or internalization, we see the difference. Now the question that you asked about the day 2 good quality and day 2 bad quality versus control, apparently you very correctly said that compared to the control and the bad quality one, the day 2 nanoparticle concentration was low, right? Now our explanation is that at that time point, the nanoparticles existing in the culture media must have been internalized. That is the kind of an idea that we have at the moment. We don't have a proof for that. That is the idea, because as I mentioned beforehand, when we supplemented EV depleted culture media for the embryo growth, beyond day 5, these embryos didn't develop. That means some critical element that is needed for the embryonic development, that is carried along with the culture media, are being utilized by the embryos at the beginning. Of course, for your information, there is something called silent embryo hypothesis, where embryos are kind of not dependent on the external environment at the very beginning, but it contradicts with what you observed with this drop in the nanoparticle concentration in the good quality embryo group compared to the control group, right? So if there's an kind of an internalization, that must have been observed only in the good quality embryos. Now it's very correctly, as you said, among these EVs, to which extent, which number of particles are EVs, which we don't know, because there's a kind of a mix of these two particles. That is the problem of EV research in embryo culture media compared to when I work with cell culture media. I have to deplete EVs from the culture media because FBS carries so much of EVs. So we had to do the filtering, which cannot be done in the embryo derived EVs. So it's a difference.
Okay, perfect. Thank you for that. And I just want to say your candidate that you tested for predicting pregnancy outcome, I those values that you gave for the positive predictive value are still excellent, because the one thing you cannot control for is the ability of the woman to actually get pregnant. The embryo might be fine. It might be something in the endometrium of the woman who is having the embryo transfer that doesn't allow that implantation to happen and successful pregnancy. So clearly a lot of great work that you've done. I'll move on to the next person who had a question, which is Sophie. So Sophie, if you'd like to unmute yourself and ask your question, please do so. Otherwise, I'm happy to ask it for you. It is in the chat box. So Sophie asked, if you take the EVs released by the good quality embryos and add them to bad quality embryos and vice versa, does anything happen to the morphology or development? Did you check this?
Well, now we had tried actually, simply it's not about taking EVs from good quality embryos to the bad quality, but what we have tried in 2019 is we culture EVs as groups. So, when the embryos are cultured in groups, their development is better because they are mutually supporting, not only through the EVs per se, but through other trophic factors which are released by EVs. These are called embryo-trophic factors. Now, the requirement was that we wanted to supplement that kind of EVs to the culture media where the embryos were cultured individually. So it's a kind of an EVs from good quality and bad quality embryo EVs, and these are kind of compared unsupplemented group with the embryonic EV supplemented group, right? So that is the same thing, but we unfortunately there was a kind of a trend, but there was no statistically significant difference. So we at some point that we had to stop that. This is a parallel study that we started, but we had to stop that in 2019 October somewhere, and it was not continued. But it was not about supplementing good embryo derived EVs to the bad embryonic EVs to support or facilitate its further growth or something. That was not what we have done.
Okay, perfect. And then the follow-up question was from Sophie again, just to say, not sure if you mentioned this during the presentation, but is the cargo different between the good and bad quality EV embryos?
Well, when I was isolating EVs from this, can you repeat the question, please?
Sure. So just, did you analyze the cargo of the good and bad quality EVs? Were they different?
I got the question, yeah. Now, honestly, from the embryonic EV point of view, we didn't manage to isolate, characterize in terms of RNA or the molecular protein levels, because at that time our understanding was that they don't have a sizable amount for that quantification, because but I know another group, a Chilean group, came up with RNA based quantification, but still it's not from a single embryo, but it's from a pooled embryo culture media. So some kind of pooling had been done there also. So if you measure individually embryo derived EVs, then apparently what we had to do was to measure them only by means of NTA. So nanoparticle tracking analysis, that was the only approach that we took at that time, because it was long delaying. Therefore, we had to finish off the work at the NTA level. But if I'm pretty sure the technology must have advanced now. The sensitivity of these assays must have advanced. So therefore, people should be able to look into this other molecular. Let me say the molecular cargo of EVs from individual embryo culture media samples.
Yeah, there's definitely improvements in technology and array-based platforms for single particle analysis that they're really going for. I think multiplexing is still proving to be a bit of an issue. So being able to perform in-depth proteomic or RNA analysis on EVs from individual embryos is probably still going to be very challenging, but something to aspire to for sure. Does anyone else have any questions they'd like to ask?
I had one question. So it's fairly simple though, but I just wanted to know, in the NT analysis, which population, respective with their sizes, were you looking for? Like, which was the most enriched population of EVs that you found and that you wanted to focus on in the entire pool of EVs that you must have gotten?
Well, now it all depends on the settings you use. because for example now in the NTA what we used was again it depends on the camera sensitivity and all, but based on the sensitivity and specificity, the sensitivity and all these parameters that we used, most of our particles are measured around the 100 nanometer size range, because if you put them together in graphs, always the peak was at the around the 100 nanometer size range. But if you change the settings, of course that shift to the right, then of course if you could have measured all these larger particles, but then again it's all what depending on what you want to measure. But surely that's why we always report the settings that we use when we are measuring these particles, because always it's not all the particles that are being measured there, so it's a kind of a snap. So that is the
Right. Great, that's all. So I think we're done with the questions now, and this was an amazing talk, first of all, because we don't really get the chance to talk often about the socio-economic challenges that our country faces. And I mean, it's still a little bit struggling in India as well, even though we have a couple of groups working on EV research specifically. But yeah, I mean, here's hoping to have a lot of good research coming out from the Asian continents, especially India, Sri Lanka, and other neighboring countries in EV research. So maybe we have an Indian Society of EVs or a Sri Lankan Society of EVs going ahead. So thank you so much for this amazing presentation. And I think, yeah, so thank you on behalf of SNEV as well. So yeah, it was lovely to have you with us.
Thank you very much for inviting me.
Thanks a lot. Okay, thanks.
Thank you.